Okay, let's start immune 4. This should be the last of the immune lectures. So, humoral immunity, effective response of B cells. And we actually already know what that is. Antibody production by plasma cells. And once that happens, we have what's called an antibody titer in our blood. That means the level of antibodies throughout dissolved in your blood throughout your body for that particular antigen. Immunoglobulins are antibodies. They have variable regions and constant regions. The variable regions are the parts that bind to the antigens. Remember, these molecules are shaped like Ys. They have 2 antigen binding sites. The constant region distinguishes the type of antibody. And there's about 5 or 6 different types. We're not going to get into all the specific types. They're all called IG something. There's IgG, there's IgA, there's IgD, there's IgA. There's a whole bunch of them. We're just concerned with the type. We just can think of them all as one type. Okay, there is a representation of an antibody. So like a Y, as I mentioned, has actually it's a 4-chain protein. 2 heavy chains, 2 light chains held together by disulfide linkages. So these are sulfur-containing amino acids. Antigen binding site as the variable region. Those are all different depending upon the antigen that it is designed to bind to. And then the base of the heavy chain is where the constant region is, and that tells you what type of antibody it is. Okay, how do antibodies work? Mechanism to render antigens harmless. So in other words, how do you just having this protein bind to this foreign antigen on a foreign cell or virus, how's that going to actually destroy it? One, neutralization, one thing, and that's just happens with the poisonous proteins. And this is the deal more like a snake bite and things like this where you serum. You neutralize the poison. It can't bind to cells and damage cells. Complement fixation, opsonization. So these antibodies bind to all these different antigens on the surface of the bacterial virus. It's called opsonization, and it activates the NK cells. The natural killer cells, that's one thing it activates. It also activates macrophages, I believe. Just a second. Okay, it's also called ADCC, antigen-dependent cellular cytotoxicity. Agglutination, they cause, and this is similar to blood typing, when the antibodies can agglutinate, red blood cells, it can agglutinate, make into clumps these bacteria or viruses. And what this does is stimulates macrophages to come along and swallow them up. Precipitation is a similar thing. It makes it fall out of solution. And and when that happens, it's also part of neutralization. These are neutralized. If viruses are agglutinated or they precipitate, they can't bind to the cells that they have receptors for. So this is how antibodies work. It might be in the next slide too. Here. Yeah, right here. Neutralization, antibodies cover biologically active portion of microbe or toxin. Agglutination, antibody cross-link cells forming a clump. And this stimulates Phagocytosis by macrophages. Precipitation, forming an insoluble antigen antibody complex in the precipitation. That falls out of solution. I think macrophages can clean those up too. Complement fixation. The variable part of the antibody attaches to the antigen in the foreign cell. The constant region of the antibody actually binds to a white blood cell. And this finds its way, or binds to complement. And this finds its way, I think, to spleen and liver and those places destroyed. I know, spleen. Opsonization. These bacterium, whatever, is covered with antibodies and it stimulates phagocytosis by a phagocyte, the neutrophil or macrophage. Then activation of natural killer cells. And this works the same way as T lymphocytes do. The secretion of perforins and granzymes to rent a big hole open in the cell and put the granzymes in and just chew it up. So here's what gives you what's called primary and secondary responses. The primary response is you first come into contact with this pathogen. And this is delayed. It takes a while. Remember, it has to travel the lymph nodes. You have to activate, and to have a B cell, a single activation first by bumping into the antigen. Then you have to have a helper T cell activate that helps the helper T cell to go through clonal selection. This takes time, days, maybe a week or more. In the meantime, you might be really feeling really lousy. But then you get over it. Now you have secondary immunity. Now, if you ever come into contact with the exact same pathogen again, you already have the memory B cells, you already have the antibodies, you can attack it immediately with no delay, and you won't even know you've come into contact with it again. Forms of adaptive immunity, as far as the antibodies goes. There could be natural immunity or artificial immunity. And there's 4 different types, adding those two together. Natural active, artificial active, natural passive versus artificial passive. Okay, natural means you make your own antibodies. All right? Natural active means you've come directly into contact with this pathogen, you've made your own antibodies, you got them through it. Now you have you're immune to it. Artificial active is you make your own antibodies, but you didn't come into contact with the pathogen itself, or in a modified form or a killed form. In other words, vaccination. You got vaccinated against this particular pathogen, and you may have only gotten part of the coat protein. I got vaccinated against hepatitis B. I only got the coat protein. So that's artificial active is vaccination. Natural passive. Passive means somebody else made the antibody, and you get it, right? But it's a natural process. And that natural process would be, only one example is breastfeeding. You can give, mom can give her a breastfeeding infant antibodies through breast milk, and these antibodies are not chewed up, they come up, they get exocytosed right to the baby's bloodstream, and they're there, right? That's natural passive. Artificial passive is, you're getting antibodies made by somebody else, but in an artificial way. And that is injection. And this is two ways. One would be the anti-sera for snake bites and spider bites. And also, the vaccine you're talking about, the antibody vaccine for the coronavirus, that's taking antibodies made by somebody else, injecting them into you. This would also be an example of artificial passive. So there's the primary response and secondary response. See the level of IgM and IgG days from that first exposure. So it's take 15, like 2 weeks almost, to get a nice titer of antibodies and wipe this thing out. So then, if you come into contact with it again, look at the secondary response. Huge numbers of antibodies made within a day or less. And you completely wipe it out, take it out, and you don't even realize you're not sick at all. Okay, the vaccinations we give, but sometimes live. Sometimes talk about, oh, it's a killed virus. Well, that's kind of a misnomer because you can't kill a virus because it's not alive. You can't kill something that's not alive, but you can deactivate it. And most vaccinations are deactivated viruses. Sometimes they're just the viral co-proteins, and sometimes they're genetically modified. That's what my vaccination to hepatitis B was. They took the gene for the hepatitis B co-protein, put it into a bacterium, grew up the bacterium, collected the protein, made it into a vaccination. But sometimes you got to use live, like the pertussis, or whooping cough vaccination. For some reason, they got to use live virus, and a small number of children will actually come down with it because it's a live virus. And also, back in the day, we were worried about polio. And polio was a scourge. Everybody was just so worried about polio. This guy named Salk came out with a polio vaccine. It worked pretty well. But then this guy named Sabin came up and said, I got a better vaccine, but mine is live virus. So then he persuaded the federal government to use his vaccination, but that means a small number of people who were actually going to come down with polio. And that sounds bad, but according to the government, it's better to have better protection for 99.9% of people and have that like 1/10 of 1% come down with the illness, but not if you're in that 0.1%, of course. Mostly a B cell response. Remember, B cells can be activated just by the dissolved antigen. So I had just the dissolved viral protein in my blood, and I made a B cell response, and I'm immune from hepatitis B. That doesn't work for T cells. Remember, T cells only work with cells, only kill foreign cells or infected cells. Okay, the disorders. In the book is a bunch of hypersensitivity disorders. I'm not going to get into those. These are allergies and asthma. An immune response, really inflammation to an innocuous protein. Some people have barely any. I have barely any. Some people have, are miserable when the pollen season is around, but it's not really serious. Some people have life-threatening allergies. They cannot come into contact with this innocuous. Well, it is innocuous for everybody else. Like peanut allergies and shellfish allergies sometimes are really dangerous. And these people can't even come in the same room with a peanut. Okay. Autoimmune diseases. Your immune system makes an error. For some reason, it thinks that this self-protein on certain cells is foreign and should be attacked. Diabetes type 1. The beta cells in a fetus, the fetal immune system thinks they're foreign and kill them. And even though the gene for insulin is in every cell in your body, it's only expressed in beta cells. That's it. So once they're gone, no more insulin. MS, multiple sclerosis. This is an odd one. It comes on sometimes out of the blue. you come down with MS. And the thought is that you came into contact with some pathogen that has a very similar protein as to the protein on myelin sheaths. And so after the virus or whatever is wiped out, then the immune system looks at their myelin sheaths and saying, oh, there's some more, let's attack it. Lupus, the same thing. Rheumatoid arthritis, the same thing. Then there's immunodeficiency. It's something called severe combined immunodeficiency or SCID. This is essentially you're born without an immune system, essentially. You don't have an immune system. And you have to be protected from every foreign organism, every virus, even though lots of them are innocuous, because you don't have an immune system. And this is what the bubble boy. Remember the bubble boy? He was kept in this big bubble, and NASA designed this bubble suit, this space suit for him, so he could go outside and play in the yard, but encased in this plastic suit. But then they found out that a way to actually cure this problem is to get a bone marrow transplant from somebody who has a very similar set of MHC type 1 proteins, cell ID proteins. And this was in its infancy. This was bone marrow transplants were in its infancy. And this is experimental. So they found out that his sister had a very close match to the cell ID protein. So they took some bone marrow from her, injected it into him, and they killed him. Because they didn't think to test for Epstein-Barr virus. 85% of us are infected with the Epstein-Barr virus. It's usually a completely innocuous virus. It's passed through saliva from the salivary glands. So if you share saliva with somebody and they're infected, you're infected. But in most people, it doesn't do anything. It's a virus that we've grown up with and is no longer dangerous, except every once in a while, it becomes activated and causes mononucleosis. And you're really sick and you're really infectious and you feel miserable, sometimes for months, but you get over it. It's not lethal. And a lot of these viruses that are called constituent viruses, are called isogenic viruses, are viruses that infect us, incorporate their genes into our genome, and we have them for life. You can never get rid of them once. This is true for all the herpes viruses. But herpes may be uncomfortable, embarrassing, but they don't kill us. And it's really good because lytic viruses kill the cells they're in. Like polio was a lytic virus. It kills the cells it's in, and it infects motor neurons. So when it makes new viral particles and kills this motor neuron, you don't get motor neurons back. And that's how people will be paralyzed by polio. And fortunately, herpes viruses also infect nerve cells, but it doesn't kill the cells it's in. These viral particles are exocytosed. And then they're attacked by the immune system and they're knocked out. That's why usually the longer you have these herpes infections, the smaller the outbreaks become, because you make up a huge clone of antibodies, and once these viral particles become active, you can get rid of most. Okay. Acquired immune deficiency. This is AIDS. It's caused by HIV, the human immunodeficiency virus that evolved from a monkey virus called the simian immunodeficiency virus, which isn't lethal in other primates. And somehow it got mutated and it became to infect humans. And it crossed to humans, not because humans were having sex with monkeys, but because we were eating monkeys and getting the blood on them and getting infected that way. And it actually is what's called retrovirus. It has RNA for its nucleic acid. It also has like a phospholipid coat. It almost looks like a cell. The cells it attacks are the helper T cells. That's really the tragic part of this. And what happens is it starts killing helper T cells. And then once you get infected, and then your body, your bone marrow just makes more helper T cells. And for years and years, you keep up. The helper T cells are knocked down, you make some more of them, but after about 7 or 8 years, your bone marrow just gets worn out. You can't make the helper T's as fast as they're being destroyed by the virus. So then the helper T cell titer drops off, and when helper T cell titer gets low enough, you start becoming susceptible to what they call opportunistic infections. Infections like nobody gets unless your immune system has been compromised, has been destroyed. Like Kaposi sarcoma and these bizarre fungal brain things that like nobody gets unless your immune system is destroyed. So it's the opportunistic infections that kill people that have untreated HIV, not the virus itself. And HIV is a really weak virus outside the body. It dies real soon. You can't get it from dry blood, just from wet blood, or it's sexually transmitted, or body fluids with a virus from an infected person. That's not true for hepatitis. You can get hepatitis from like completely dry blood. Not true for HIV. Okay. But with the development of drugs, HIV infection has become a manageable illness, and taking the drugs for your whole life on a very regimented way will keep you almost virus-free and keep you alive. I can say it because he's a celebrity, Magic Johnson. He got infected a very long time ago, and he is fine because he's taking these drugs and has taken them for the whole time since he's been infected. Okay, the drugs that work. There's a few drugs, and the reason we give them a cocktail of drugs is you don't want to give one drug because just like penicillin, nobody takes penicillin anymore because the bacteria become immune to it. Viruses can become immune also to drugs if you just use one. Now, there's a cocktail of drugs. One is the artificial nucleotide. I think it's artificial guanine that the virus takes and incorporates it into its nucleic acid, and it doesn't work. Actually, there's an anti-herpes pill that actually does the same thing. There's a transcriptase inhibitor. Oh, here's the problem. Oh, I forgot about this. Here's the thing. HIV has a problem, or any RNA virus has a problem replicating in our body. Why? Because it has to replicate RNA. Well, we don't do that in our bodies. We don't make RNA from RNA. We make RNA from DNA, not RNA. We don't have the enzyme for that. So how is HIV going to replicate its RNA? So what the virus has is an enzyme called reverse transcriptase that replicates DNA from RNA, which we don't do. We make DNA from DNA. We don't make DNA from RNA. But the viral enzyme in the viral particle that does make DNA from the RNA, then it can use that DNA to make more of its own RNA. And then they hijack our machinery, our RNA transcriptase to make its viral RNA. Well, they have a drug now that inhibits that reverse transcriptase and prevent the DNA being made from the viral RNA. And finally, peptidase inhibitor. This is the latest one. It's strange. In our bodies, we make, when we translate an RNA, we make one protein from it. That's it. But the viral RNA is like this huge viral messenger RNA. It's this huge thing that actually has 3 proteins. It makes a huge, like, triumvirate protein, gigantic protein that has 3 parts. And then a viral enzyme has to chew this thing into 3 parts, and then these viral proteins become active. Well, there's a drug that will inhibit that peptidase. And so the viral protein stays these big conglomeration of 3 proteins that cannot work because they have to be separated in order to work. And that's the latest one. And that's the one that really has done a job and made HIV infection a manageable illness now. And that's it. That's immune4. So we're through with the immune system. So let me put this up, and we'll be A-OK. See you later with a different chapter. All right, where is it? Oh, here it is.